Thanks for the introduction, and hello everyone. I'm Su from Microfluidics, and I'd like to thank the conference organizer again for giving me this great opportunity to present you the scalable microfluidizer technology and how it can be applied in drug delivering nanoparticle manufacturing processes. Here's the agenda for my today's presentation. I'd like to start with a quick introduction to tell you a little bit more about Microfluidics as a company, and briefly talk about drug delivery nanoparticles and some common challenges and requirements during the development and production of those nanoparticles. And then talk about why Microfluidics microfluidizer technology is a unique technology and the ideal solution, with a few case studies to demonstrate its capabilities. I will end my presentation by showing you our diverse product portfolio. I do want to apologize that unfortunately I'm not able to attend the live Q&A session after my presentation, but you'll find my contact information again at the end of the presentation, and please feel free to reach out to me with any questions or comments. Microfluidics is under our parent company, IDEX. IDEX groups all business units into 3 different segments, and we're part of the Material Processing Technologies, or MPT platform, a division under the Health and Science segment. The platform is constantly growing and now has 5 brand names: Microfluidics, which is where I'm from, Fitzpatrick, Metcon, Quadro Engineering, and Stardose. Microfluidics itself was founded exactly 40 years ago, so this year is our 40th anniversary. And we specialize in design and producing high-shear fluid processors using our unique interaction chamber technology. We're located in Massachusetts, just outside of Boston, and have a global presence with customers from all over the world. As I mentioned already, we're part of the IDEX MPT platform. Our technology serves many industries, primarily the pharmaceutical and biotech industry, but are also widely used in other industries such as chemical, food, and nutraceutical, as well as cosmetics, in both development stage and commercial scale manufacturing. With our extensive experience in applications as well as machine design, especially for cGMP compliance, we can provide tailored solutions to enable you achieve your specific processing goals. I think everyone knows very well that conventional drug delivery methods face a lot of challenges, including low solubility, which leads to low bioavailability, poor in vivo stability, and adverse side effects, etc. Nanotechnology is a fast developing area in recent years, which has led to the development and applications of a variety of nanoparticle-based delivery systems. Such systems offer many benefits comparing to conventional large-sized materials, which I've listed a few, but I'm not going to read through them here. So whether you're working on the next exciting drug or treatment or trying to repurpose existing molecules, keep in mind that a good delivery system can also help in many different ways. A few popular and successful nanoparticles as drug delivery platforms are shown here as example, and I'm going to be covering most of them later with my case studies. I'm talking about developing and manufacturing drug products, including drug delivery nanoparticles. There are many factors or key quality attributes needs to be considered. The first and foremost are the physical chemical properties. Among so many important properties, particle size characterization is one of them and is arguably the most important one, as they can significantly affect the stability of any colloid system and its in vitro as well as in vivo performance. So to be able to control the size and distribution of nanoparticles is critical. One more thing I'd like to point out here is that the particle size and distribution are also very important for generic drug development, because particle size and distribution is often the key criterion in that demonstrating bioequivalence. Next requirement is the selected technology should be robust enough and be able to produce repeatable results to ensure batch-to-batch consistency. The tray should also be future-proof in terms of scalability, which means the process needs to be easily and directly scaled up for manufacturing with guaranteed results. Another requirement, or rather benefit, is the process should be able to improve or lead to easier downstream or adjacent processes. For example, injectable drugs, require sterilization to remove bacteria. Although autoclave is a widely used sterilization process, it is not often feasible for heat-sensitive formulations. A simple and more economic alternative way to do this is by utilizing stereofiltrations. Stereofilters are made of membranes, have rated pore size of 0.22 microns or 220 nanometers, which implies that the majority of the particles in the product should be smaller than this size to ensure an efficient filtration process. Last but not least, the manufacturing equipment needs to allow the entire manufacturing process to comply with regulatory requirements such as cGMP. Many pharmaceutical manufacturing processes also require the equipment is capable of running CIP and/or SIP processes. Microfluidics technology can actually address all these challenges and requirements from development to production, with its unique advantages in achieving target processing results and guaranteed scalability. The principle of microfluidizer technology and how the system works is illustrated in this diagram. Product with large particles enters the system via the inlet and is pulled into a constant pressure pumping system, which pushes the material through a fixed geometry injection chamber at pressures up to 30,000 psi or 2,000 bars, where the material experiences consistent high shear rates and impact forces. All material receives the exact same treatment to achieve consistent and reliable results. After passing through the injection chamber, the product temperature can be efficiently controlled if needed. Microfluidics offers two different injection chamber designs. As shown in the top right graph here is the white type chamber. The white chamber has two microchannels and the sample impinges on each other after passing through the channels, and is ideal for preparing most drug delivery nanoparticles such as nanoemulsions, liposomes, lipid nanoparticles, protein-based nanoparticles, etc. The bottom graph shows the Z-type chamber. The Z chamber only has one microchannel and is ideal for processing samples containing solids, including cell disruptions. Before I move on to the case studies, let me quickly explain how do we scale up and guarantee the results. Unlike other conventional technologies, when we scale up our system, we don't change the size of the microchannels. Instead, we use multiple parallel aligned microchannels with same size. As long as other parameters are kept the same, the product will experience identical uniform shear and impact treatment inside each channel, and that's achieve guaranteed linear scale-up results. This led to my first application, nanoemulsions. I will give an example of a drug-carrying nanoemulsion that was developed and produced with the microfluidizer technology. This is based on the research published by Professor Mansour Amiji's group at Northeastern University. The aim of this study was to design a delivery system to carry widely used chemotherapeutic agents, toxins, and be able to deliver the drug to the tumor site. The drug of interest is a new generation toxins developed with high potent and excellent efficacy. To further reduce the toxicity and increase the retention in the tumor, the toxin is also conjugated with DHA. Due to the lipophilic nature of the conjugate, all-in-water emulsion is an ideal delivery platform. To achieve desired efficacy and target delivery, the final emulsion will be ideally within a size range of 100 to 250 nanometers. To prepare such emulsion, researchers used the microfluidizer processor and was able to successfully produce the nanoemulsion, as can be seen from these graphs. The resulting nanoemulsion had a unimodal distribution with an average particle size of around 200 nanometers and a polydispersity index or PDI of around 0.1. Usually, colloid system with a PDI below 0.2 is considered uniform. So in this case, the produced nanoemulsion had a very narrow particle size distribution. The emulsion also had a desired negative surface charge to help prevent aggregations. Finally, encapsulating conjugates did not seem to affect the particle size at all. Analysis also confirmed that almost 100% of drug loading efficiency was achieved. Small particle size and narrow distribution also helped the stability of the produced nanoemulsions. As shown here, the emulsions were stable for at least a couple months under all storage conditions. The best stability was obtained with the storing at 4 degrees Celsius, which showed excellent stability during the 6 months duration of the study. Additionally, under all storage conditions, the nanoemulsion formulation was able to maintain the drug encapsulation level, while the free drug solution showed significant reductions at much higher speed. When intravenously administered, the nanoemulsion showed diffused pattern in naive mice and gradually decreased over the time and almost cleared out within 24 hours, while nanoemulsion in tumor-bearing mice induced a massive signal in the peripheral of the tumor region and become more concentrated over the time and persisted over 24 hours. The plasma pharmacokinetic data showed that the nanoemulsion formulation significantly increased the plasma exposure with almost a 6-fold increase in tumor-bearing mice, and the targeting efficiency was almost doubled with the nanoemulsion formulation. The next application is liposome. Liposomes have been used as both drug delivery systems and vaccine adjuvants in commercialized products. In this case study, I'll be showing you some data from our collaboration with Professor Yvonne Perry's group at the University of Strathclyde. We were interested in another class of cancer drugs, doxorubicin. Doxorubicin is a well-known anti-cancer drug that has been approved for treating several different kinds of cancers and tumors. Liposomal doxorubicin is formulated with pegylated lipid and was the first FDA-approved nano-drug delivery system based on pegylated liposome technology. It is currently available as Doxo in the US market. The aim of this study was to develop a better process to produce generic version of doxorubicin liposome formulation with the ease of manufacturability and scalability. Obviously, as mentioned before, the process needs to be able to achieve target size range, which is between 80 and 120 nanometers in this case, and high drug encapsulation rate of over 90%. Another goal of this study was to see if the liposomes can be prepared via a greener way, meaning without using any solvent at all. This is important because so far solvents such as chloroforms or ethanols have to be used either sooner or later during the manufacturing process, regardless of method of selection. The use of solvent is not preferred due to their potential safety and health concerns. So we want to see if the microfluidizer technology can be used to create liposomes without using any solvent. And we've successfully achieved this goal. The new process illustrated in this diagram for making doxorubicin liposomes. You can see instead of forming lipid fumes, we simply hydrate all lipid ingredients directly in the aqueous buffer and then use the microfluidizer processor to downsize the vesicles. Also, empty liposome vesicles were created first. Doxorubicin was loaded into the aqueous core inside the liposomes via the remote loading technique afterwards. To do this, a buffer exchange step is included after the size reduction step to create a pH gradient for drug loading. Finally, the liposome solution went through additional purification and sterilization steps. Apologize for this busy slide, but the charts show the particle size, PDI, surface charging, and drug loading profiles throughout the entire manufacturing processes as shown in the previous slide. Results show that the desired particle properties can be achieved by processing at 2 different pressures of either 18,000 psi or 20,000 psi for just 2 or 3 passes. Both higher pressure and more passes resulted slightly smaller particle size and PDI. Particle size, PDI, surface charge, and encapsulation efficiency remained fairly constant throughout the entire manufacturing processes. The reason for the change in the zeta potential was due to the change in the pH, which is required for drug loading, as mentioned before. After loading, the net surface charge went from neutral to significantly negative charged and stayed constant afterwards. We then compared the liposome created with the new solvent-free method to the conventional solvent-based method. Please note here that although the microfluidizer processor is definitely capable of making liposomes via the traditional way, for comparison purpose, the sonication method was used in this case. The solvent-free method not only proved its capability, but also yielded overall better results, as the encapsulation efficiency was slightly higher. The cryo-TEM images also revealed that the sonication method produced the non-uniform vesicles, whereas the microfluidizer processor was able to create uniform small unilaminar vesicles. So this confirms that the microfluidizer technology is not only suitable for producing liposomes, but also superior to other competitive technologies. In addition to this example of encapsulation hydrophilic actives with liposomes, encapsulation of hydrophobic API, amphotericin B, which was used as a model drug using liposomes, was also demonstrated, and more results can be found in the paper that has been recently published. The next application is lipid nanoparticle. The recent and still ongoing pandemic has made those particles famous due to their ability to carry and deliver nucleic acids, such as DNAs and RNAs. Naked DNA or RNA are highly unstable, and when delivered by itself, is rapidly degraded after injection under physiological conditions. Therefore, proper delivery systems are required. Among several different approaches, lipid nanoparticles are by far one of the most attractive and successful delivery platforms. This case study is based on results published recently by HDT Bio in collaboration with a few other research institutes. In this case study, a lipid inorganic nanoparticle was developed for delivering replicon RNA-based COVID-19 vaccine candidate. Unlike the approach used by some of the current RNA LNP-based COVID vaccines, the approach taken here has two major differences. The first is the use of rep-RNA instead of mRNA. Rep-RNA provides increased antigen payload due to its replicative nature, which lead to more robust immune responses even after just a single dose, whereas mRNA typically requires multiple and orders of magnitude higher doses. The second is how the RNA LNP system is prepared. Conventional approach prepared the RNA LNP system within a single step. The approach works well. However, it is a major issue of using large amount of solvent and stability. It is believed that RNA is the component that affects the overall stability. That's why all current RNA-based vaccine requires extreme cold storage conditions, but still have very limited shelf life. The LNP systems, on the other hand, are usually much more stable. That's why a two-step approach was adopted by the researchers in this study. As illustrated here, the LNP was manufactured first without any payload, meaning just the blank LNP particles. And the rep-RNA was simply added and conjugated to the LNP just prior to immunization. As can be seen here, blank LNPs created by a microfluidizer processor achieved a very small particle size of 50 nanometers and a very narrow distribution. After loading the rep-RNA, the particle size grew slightly, which was also an indication of a successful RNA loading. Particle size monitoring of the blank LNPs showed they were stable for at least 12 weeks, even under room temperature condition. The RNA LNP complexes also showed good stability as particle size remained constant for at least 2 weeks at either room temperature or refrigerated condition. In addition, rep-RNA also remained full integrity as confirmed by gel electrophoresis. Just quickly go through some results regarding the immunogenicity of this vaccine formulation. Data showed that both mouse models achieved robust antibody responses and much higher responses after the booster immunization for all doses and age groups. Also, the induced immune responses seemed to be the Th1 biased, as indicated by the higher IgG2 response. Interferon gamma responses in spleens and lungs showed robust T-cell responses for all those groups. Finally, I'd like to point out that although in this case, the RNA LNP complex was used to formulate vaccines for infectious disease, the same concept can be used for cancer vaccines as well. But this approach is not just limited to vaccines. Using LNPs to deliver RNA-based drugs also has great potentials in tumor therapeutics. The last application in my presentation is different from all previous nanoparticles because this is protein-based nanoparticles. Specifically, I'll be talking about albumin-based nanoparticles. Human serum albumin is the most abundant protein found in the human body, so it's biocompatible, biodegradable, and non-immunogenic. HSA also has multiple binding sites which can bind to either hydrophobic or hydrophilic therapeutic drugs. Most importantly, albumin can utilize the GP60 albumin receptor and spark protein pathway to effectively deliver albumin-bound therapeutics to tumor cells. All these advantages make HSA nanoparticle an ideal platform for delivering anti-cancer drugs that are high affinity for albumin. Paclitaxel, a well-known and widely used commercially available chemotherapeutics agent, is one of such drugs. The drug albumin complex is formed via the unique nanoparticle albumin bound, or NAB technology. Abraxane is the first FDA-approved commercial drug which is manufactured using the NAB paclitaxel technology. The case study presented here is based on research published by researchers at several research institutes from China. It was aiming to compare two different forms of NAB paclitaxel. One is non-cross-linked, which is similar to Abraxane, and the other is cross-linked version. Non-cross-linked NAB paclitaxel nanoparticles were prepared via the emulsion evaporation method. Basically, a paclitaxel containing solvent is emulsified in albumin solution, which was done by the microfluidizer processor. After forming the desired nanoparticles, the solvent is subsequently removed to allow forming the solid NAB paclitaxel nanoparticles. The cross-linking version was obtained by adding additional cross-linking agent to the non-cross-linked nanoparticles. The reason of doing the additional cross-linking step is some researchers believe that this will help improve the stability of the particles. As can be seen from both particle size analysis and TEM images here, non-cross-linked NAB nanoparticle was successfully produced with the microfluidizer technology with desired particle size and morphology similar to Abraxane. The mean particle size was around 120 nanometers with a very narrow distribution of PD at less than 0.1. Results showed that cross-linking had no effect on the particle size and size distribution. Also, both versions achieved a very high drug encapsulation efficiency of over 95%. This slide shows the biodistribution of paclitaxel and the anti-tumor efficacy. You can see that based on the AUC data, the biodistribution of the cross-linked and non-cross-linked nanoparticle formulations were slightly different. But more importantly, non-cross-linked nanoparticles rapidly accumulated into tumor, which indicated an enhanced tumor targeting efficacy comparing to the cross-linked particles. This was also confirmed with the AUC data. The use of non-cross-linked particles increased the percentage of drug distribution to the tumor by a factor of 2. The enhanced targeting efficiency led to better therapeutic results, as shown in this plot. Although both nanoparticle formulation could effectively inhibit tumor growth compared with the control group, the non-cross-linked formulation was more therapeutically active and slowed down the tumor growth considerably. I hope from the applications I presented to you today, you were able to learn more about microfluidizer technology and are convinced that this technology is the ideal solution for manufacturing drug delivery nanoparticles. Before I conclude my presentation, I want to show you quickly our diverse product lines. The top are our lab units, and the bottom are our pilot and production machines. For all the pilot and production machines, and plus this one lab unit, we also offer biopharma version that are in compliance with cGMP requirements. And with that, I'd like to thank everyone again for your attention. As I mentioned earlier, I apologize again that I'm not here today to answer your questions live. However, our local agent, Animed, has a virtual booth here at the conference, so please make sure to stop by and find out more about the microfluidizer technology and ask any questions you may have. You can also reach out either to me or to Animed with our email address listed at the bottom. Thanks again, and hope you enjoy the rest of the conference.